Hi, this training video will demonstrate two techniques commonly used in microbiology experiments. The first is streaking agar plates to grow single colonies from a culture broth. The second is testing the susceptibility of the bacteria to certain chemicals by using small paper discs coated in antibiotic. Since we're using Bunsen burners, make sure you watch the training video on Bunsen burners found on the ISI YouTube channel. For safety, you have to wear your lab coat, safety goggles, and gloves, and use Dettol or a similar antiseptic to spray down your work area before and after your experiment. When you're working with agar plates, label the bottom of the plate with the type of bacteria, the date, and your initials. You don't want to label the lid because lids move around and they may get mixed up between plates. This is an inoculating loop, and it consists of a long handle and a wire with a loop at the end. This particular model is calibrated to pick up 10 microliters of fluid each time. As described in the Bunsen burner training video, the hottest part of the flame is found at the tip of the blue flame. So when you want to sterilize your loop, place it in that area until it glows bright orange. And when you're done, you don't want to pick up your bacteria right away because the heat will kill any of the bacteria. So you want to wait about 15 or 20 seconds until it cools down. You can test this by touching a clean area in the agar plate. As long as it doesn't hiss and melt the agar, that means that the loop is cool. When you're ready to pick up your bacteria, use your pinky finger to hold onto the lid, remove the container, and place the wire loop in the culture broth and draw out your 10 microliters. Replace the lid, and now you're ready to streak. Streaking an agar plate involves diluting the initial amount of fluid picked up by the inoculating loop until single colonies grow. The first streak, which contains all of the fluid picked up, will be spread out on the plate. Now, too much bacteria will grow here, so you won't see the individual colonies. The second streak will pick up some of the bacteria, the first streak, spread it out and dilute it. The third streak will dilute it even further by picking up some of the second streak and spreading it out until single colonies grow. The way this is done with an agar plate is to flame your loop, pick up the bacteria, remove the lid, and do your first streak. You want to make sure that you flame the loop again, wait for it to cool down, and then do your second streak, which is diluting the first streak. Flame again to kill all the bacteria. When it's cooled down, do the third streak, which should dilute it out until single colonies grow. When you're done, you want to put your plate in the incubator with the bottom side up so that any condensation that might have formed on the lid doesn't fall onto the agar. After it's been in the incubator for about 24 hours, you take it out and you should see the growth from your streaks. So the first streak is very thick with growth, the second streak is a little more dilute, and finally by the third streak, we see single colonies. When you want to test the susceptibility of a bacteria to certain antibiotics, use these small paper discs that are coated with the antibiotic. In this case, these two discs are coated with penicillin or chloramphenicol. In this case, we don't want to grow single colonies, but we want our bacteria to grow over the entire surface of the agar plate. We're going to use these cell spreaders to help with that. We're also going to use micropipettes. If you haven't used micropipette before, make sure you watch the training video on micropipettes found on the ISI YouTube channel. This one's set to 100 microliters. We're going to get a sterile tip on the end. We're going to pick up 100 microliters for bacteria, which in this case is E. coli. And we're going to put it in the middle of our agar plate. Then take our cell spreader, kind of like a rake, just going to rotate the plate around until the bacteria is spread out evenly over the surface. When that's done, we're going to use these forceps to pick up these discs and to place them on the agar plate, fair distance apart. I'm going to put our lid on. This time when it goes in the incubator, we're going to put it with the agar on the bottom because we don't want the discs to fall off if we put the agar on the top. After about 24 hours of growth, you should see that the E. coli has grown over the entire surface, and where the antibiotic has inhibited growth, there's a circle or a zone of inhibition that you can circle and measure. After watching this video, you should have a better understanding on how to streak out agar plates to grow single colonies and how to test for susceptibility using paper discs. When you get to the lab, if you have any questions, make sure you ask for help. And remember, every experiment is an opportunity to learn and develop skills no matter the outcome.